Okay, so let us just go and have a look at what we had studied in earlier class. We studied about what are gametes, what is human egg and sperm. This is the female gamete, male gamete, and how these primordial germ cells are originating. We studied that they are developed from primitive streak. They enter into dorsal wall of the yolk sac. From the dorsal wall of the yolk sac, they migrate into the gonads. Okay, gonads are testis and ovary, and the cells are migrating into the gonads, right, in the developmental stage. Why this migration is important? Because if they are not migrating properly and they are accumulating somewhere else, they can give rise to teratomas. Teratomas are embryological cancers. So we also studied about what is inheritance, how the genetic recombination is happening in male and female cells, giving rise to entirely new organism, giving rise to entirely new genetic composition, right? We studied about that. So we also studied how these primordial germ cells are dividing, and we had studied what is the difference between female and male divisions. Okay, in female we had seen that there is unequal division, okay, unequal division of cytoplasm, giving rise to only one secondary oocyte and one mature oocyte, resulting of two mitotic divisions. Okay, sorry, meiotic division. Completion of one meiotic division gives rise to only one mature oocyte, and the rest are all polar bodies. Whereas in male, equal division of cytoplasm takes place, giving rise to four same type of spermatids, okay, without any difference. So this is what we had seen, difference between male and female. Now, why these divisions are important? Because we need to understand about chromosomal disorders. Any kind of abnormality in exchange of chromosomes can give rise to trisomies or monosomy. That means extra number of chromosomes can give to trisomy. One extra chromosome can give a drastic defect, okay? Whereas monosomy also can give rise to a big defects, okay? Anencephaly and all. Okay, right? So Down syndrome is one of a chromosomal disorder where we have got, see, this is a karyotyping plate. We know that when the chromosomes are arranged in an order, we can see 21st number of chromosome we have. Instead of 2, we have got 3. Okay, so this is a trisomy number 21, Down syndrome. Okay, so this is what we try to get when we are looking at differences. Now let us understand, see, oogenesis, what happens. We have already seen that initially we have a primordial germ cell. It undergoes mitotic division, gives rise to oogonia. And this oogonia again undergoes mitotic division, gives rise to primary oocytes in prophase. This is what we had studied in the last class. And how many number of cells are there? Lot of divisions takes place when the child is a female fetus. Okay, when a child is still a female fetus, at that time the number of eggs present in her is starting at 3 to 6 weeks of gestation, it is just 10,000. By the number of 20 to 40 weeks, it increases to many number of millions, and then slowly by birth and puberty it comes to 3 lakh numbers, and by reproductive years only 400 to 500 eggs are present. Okay, now this is called as ovarian cycle. Okay, what is ovarian cycle? In ovarian female reproductive cycles we will study again later. Okay, what you have to remember is primordial oocyte undergoes into again first meiotic division already started, but it did not complete. So when the female enters into first menstrual cycle, or every menstrual cycle, what happens? 2 to 3 oocytes enter into oogenesis. Okay, so out of 2 3, what happens is only one undergoes complete ovulation. Here there is ovulation happening. So that ovulation happens only at the completion. Only one egg undergoes ovulation. 2 to 3 undergoes maturation, but only one completes ovulation. That's why only one egg is released. Okay, So this is the cycle which is followed. Okay, what are the stages we had seen? We already know that this is the first meiotic division. It undergoes first meiotic division, prophase 1, metaphase 1, anaphase 1, telophase 1. And at the end of telophase 1, what happens? Ovulation completes. Okay, we get secondary oocyte. Now secondary oocyte enter into and first polar body. This is the first polar body. Polar body is absorbed by the fallopian tubes. Okay, or it is regressed or it is apoptized. What is left? Only secondary oocyte. Now secondary oocyte, it enters into metaphase stage but will not complete metaphase stage. Why it will not complete metaphase stage? Because it will complete only metaphase stage only after fertilization. Okay, fertilization means what? Only when you have sperm entering into the egg, this metaphase stage completes and gives rise to complete mature oocyte. Okay, this mature oocyte formation will take place only after completion of fertilization. All of you are clear with this, everyone?
Yes, ma'am.
Yes, ma'am.
Right. Now this is the ovarian cycle. In ovarian cycle, what are the stages we will see? Stage 1, primordial follicle. Then you go to primary follicle. What is happening here? What is happening? The cells which are present around it they are enlarged. Okay, this is the oocyte which is inside. Okay, and above that, these are follicular cells. Where are these follicular cells coming from? They are coming from the ovary. Okay, they are coming from the ovary. They accumulate around this oocyte. Oocyte is inside. Ovarian cycle is only the entire follicular changes. Now we had seen this one. What is this? Oogenesis. Oogenesis is happening inside the oocyte. Are you all clear with me? What did I say? Oogenesis is
Yes,
happening
ma'am.
inside the oocyte. Now this is ovarian cycle. Ovarian cycle is cycle which contains follicular changes. What are the follicular changes happening? First we have primordial follicle. What is primordial follicle? Oocyte plus the follicular cells which are in simple squamous epithelium. Okay, later on when it enters into maturity, that means what? Now the girl is entering into menstrual phase. When menstrual phase comes, that is 2 to 3 follicles enter into maturity, that is entering into next stage of formation. Okay, so this is primary follicle. What is the difference between primordial follicle and primary follicle? Here the follicular cells are simple squamous. Here they become cuboidal and later on they become columnar. Okay, so that is the primary follicle. Now these cells slowly they will start dividing. Only follicular cells start dividing. Okay, oocyte is inside, right? So now these cells, when they divide in multiple numbers, 2 to 3 layers are formed. These cells are now called as granulosa cells. Okay, what are these called as? Granulosa cells. Why granulosa cells are important? Why these are called as granulosa cells? Because they will start secreting a pigment, a protein, okay, which is covering outside the follicle. Okay, it is giving a separate follicular appearance inside the ovary. Now also, if you are looking with an um, endoscope, okay, at the ovary, you can see that so many follicles are there. You see a histology slide of ovary, you see number of follicles. How you can demarcate that this is a follicle? You can see a nice outer covering of the follicle. What is that follicular covering called as? This is called as theca. What is that called as? Theca. Okay, theca means layer, covering. What is this? Theca, theca, theca. Okay, now there are two layers for this theca. Okay, theca externa and theca interna. Okay, theca externa is outer covering, which is more or less fibrous. Theca interna is cellular. It it contains cells. It contains cells which support these granulosa cells. Okay, so that is the first change which is happening in this secondary follicle. Now this is no more a primary follicle, it became secondary follicle. Secondary follicle is this. Okay, so when you see secondary follicles, what are the major changes you will see? First, there are number of granulosa cells. You will see theca externa and theca interna, and you will see a specialized protein covering, okay, outside the oocyte. Now, what is this protein covering? Called as zona pellucida. Very important structure. A special short note question can be asked here. What is it called as? Zona pellucida. What is zona pellucida? It is a protein coat which is secreted both by the granulosa cells as well as the oocyte. Now, why it is important? It forms a boundary between oocyte and granulosa cells. Why these granulosa cells are present here? They provide nutrition to the oocyte, okay, growing oocyte. So they provide channels. This protein coat gives number of protein channels, okay, gates, pathways, so that the granulosa cells are giving nutrition to the the oocyte, okay, primary oocyte. Now this is the secondary follicle. Clear all of you? How did the change happens? Primordial follicle, secondary primary follicle, then it goes to secondary follicle. Secondary follicle undergoes enlargement, gives rise to next stage, that is
Okay, ma'am.
yes. Any question? Can you all hear me?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Yes, ma'am. Yes, ma'am. Yes, ma'am.
Okay. So primordial follicle, primary follicle, secondary follicle, and we have tertiary follicle. Okay. Tertiary follicle, what does happen? This is also called as Graafian follicle. What is it called as? Graafian follicle. Now, Graafian follicle contains what? Slowly, what happens? The fluids from the ovary and the uterus, they start accumulating inside, and they push all the cells to corners, giving rise to a big space. Okay, so space is filled with liquid. See here, the space is now enlarged, pushing the entire follicular cells or the granulosa cells into outer covering and inner covering. Okay, so this is called as mature Graafian follicle. This mature Graafian follicle, a short note question, can come as a short note question. What does it contain? Outer coat of theca externa, theca interna. Okay, follicular cells. Follicular cells are now split into two layers. What are the two layers? The one which is outside, it is called as membrana granulosa. What is it called as? Membrana granulosa. Granular cells forming like a membrane. This is called as membrana granulosa. Okay. Now, some of the cells are around this oocyte. Okay, this is first meiotic division completed. Okay, so now this is a secondary oocyte. It is about to ovulate now. So this particular cells which are around the secondary oocyte, this is called as cumulus oophoricus. Cumulus means surrounding. Oophoricus, oocyte. Okay, this is called as cumulus oophoricus. This is called as membrana granulosa. Now these two layers are adherent at one place, this region. This is called as discus proligerus. What is it called as? Discus, D-I-S-C-U-S, discus proligerus. Okay, Proligerus, proligerus, discus proligerus. Okay, so discus proligerus is the area where proliferation of the cells takes place, and keep on accumulating around the oocyte and the membrana granulosa. Okay. Now this space is called as antrum. You can see that this space is called as antrum. Okay. Now the antrum is filled with liquid. What is this liquid? This liquid is called as liquor folliculi. Okay. Liquor folliculi or antral fluid. Then we have secondary oocyte. So this is a short note question. You have to draw the diagram and you have to explain each layer. Okay, how it is formed and what is its function. Clear? So what are the layers? Outside we have theca externa, theca interna. Theca externa is a fibrous layer which separates the follicle from the rest of the area. Theca interna is a cellular layer. It secretes estrogen. Okay, theca interna secretes estrogen. It maintains this follicle development. Then we have got membrana granulosa and what is this one? Cumulus oophoricus, and this combined area is called as discus proligerus. Okay, that is related to follicular cells. Then we have a cavity which is called as antrum. Cavity is filled with antral folliculi or antral liquid. It is also called as liquor folliculi. Okay, and of course the secondary oocyte after first meiotic division. So this is the stage coming from where? Primordial follicle, primary follicle, secondary follicles, okay, tertiary follicle, Graafian follicle. Okay. Now what happens at the 14th day of menstrual cycle? There is a big pressure. You can see that the outer coating of the ovary becomes very thin here. You can see a rupture of this ovarian membrane also. And you can see with a plop, okay, what happens? With a pressure, this liquid pushes this secondary oocyte outside. Okay. So it's a kind of popping of an egg. Okay. So it pops out, rest of the cells remain here except that oocyte, secondary oocyte, and few cells accumulating around it. This cumulus oophoricus are taken outside. Okay, this process is called as ovulation. When does it happen? It happens on 14th day of menstrual cycle in a 28-day cycle. In a 28-day cycle, 14th day is the ovulation day. Okay. After that, what is the rest? What is the rest of the features? In rest of the features, what you will see? These cells, they will start accumulating yellowish pigment called as lutein protein. Okay, lutein, Lutein, lutein protein. So this will become yellowish body. This is called as, now this body, when we say body, it is called as corpora. Okay, when corpora means body, this is called as corpus luteum. Luteum is lutein protein. It is accumulating lutein protein. So this product is called as corpus luteum. Okay. Now corpus luteum can be a short note question. Why short note question? How is it formed? Now we know how is it formed. Okay. What is its fate? What is its fate? Its fate is depending upon fertilization. If there is fertilization happening, this will stay for a period of 3 to 6 months and secrete progesterone. Progesterone hormone is secreted by this. Why progesterone is important? That progesterone hormone maintains the thickness of uterus, okay, for the implantation of the baby. So that's why that progesterone hormone is important, and that's why corpus luteum is important. So that is called as corpus luteum of pregnancy, that remains for where about 2 to 3 months of pregnancy. It remains there. If there is no fertilization, what will happen? This corpus luteum regresses after 7 to 8 days. Okay, because 14th day menstruation is happening. After 7 days, it slowly regresses and becomes a white color body. This is called as corpus albicans. Albican means white color. So it becomes corpus albicans. and completely engulfed by macrophages of the ovary. Okay, it gets removed off. So this is the ovarian cycle. So that time, if it is converting into corpus albicans without undergoing into maturation or lutein protein accumulation for a period of 2 to 3 months, okay, or 3 to 6 months, then this is called as corpus luteum of menstruation. The lifespan is 7 to 14 days only. Okay, so we have corpus luteum to flow chart. You can make a flow chart: corpus luteum formation and what is the fate. Fate, if fertilization occurs, then it is called as corpus luteum of pregnancy. If fertilization does not occur, it is called as corpus luteum of of menstruation. Okay, what is the time limit? In pregnancy, it lives for 3 to 6 months. What is the function? It secretes progesterone for the maintenance of the pregnancy. Okay, if there is no fertilization, then it is called as corpus luteum of menstruation. What is the lifetime? It lives for 7 to 14 days and then regresses to corpus albicans. Till here, everyone clear?
Yes.
Yes, ma'am.
Now let us go and have a look at spermatogenesis. In spermatogenesis, what happens? Again, see here, this is a testis. Inside the testis, one second, this is the testis. Inside the testis, lot of sections are there. In lot of sections, we have seminiferous tubules. Okay, these seminiferous tubules are tube-like structures. When you take a cut section of this tube and enlarge this and see, you will see that there are lots of generations of cells. Okay, from puberty stage to the almost 70 to 80 years of life. Even after that, it is a lifetime. Spermatogenesis can happen in males. Okay, that is the difference between females and males. In females, the fertility period is still 50 to 60 years of age, whereas in males it continues beyond 70 to 80 years. Okay, so if you see this wall of the seminiferous tubule, you will see generations of the cells. What kind of generations of cells? Initially we have germinal cells. We know that spermatogonia, remember that spermatogonia, they divide by mitosis and gives rise to spermatocytes. Okay, these spermatocytes divide by first meiotic division and gives rise to secondary spermatocytes. These secondary spermatocytes undergo second meiotic division and gives 4 spermatids, which are circular. Please remember that. These are circular. Spermatids are circular. Now, these spermatids undergo a specialized process called as spermiogenesis. Okay, what is it called as? Spermiogenesis, to convert into this particular shape. Okay, this process is called as spermiogenesis. This is called as spermatozoa or mature sperm. This happens only after this particular process. Okay, till if a question comes spermatogenesis as a short note question, you are supposed to write all these layers and also you are supposed to write spermiogenesis, or a separate short note question can come only on spermiogenesis. So you are supposed to write how the spermatids are converting into spermatozoa. Okay, clear everyone?
Yes, ma'am.
Yes, ma'am.
So see here how the spermatids. So how initially we have spermatogonia. These are called as type A dark spermatogonia. These appear as stem cells. Now these will divide into pale type A spermatogonia. Gives rise to, these are all mitotic divisions. Okay, they are still connected to each other. Please see that they are not separated, they are all connected to each other. Then finally these type A pale also remain as stem cells. Now they will give rise to type B spermatogonia. These are more differentiated. Once type B spermatogonia are there, now we know that yes, these are the cells which are supposed to go into meiotic divisions. Okay. So they undergo into meiotic division and give rise to primary spermatocytes and secondary spermatocytes. Till they are all connected to each other. See here, cell membranes are connected to each other. Later on, undergoes second meiotic division and gives early spermatids. Now, what is the difference between early spermatids and late spermatids? The spermiogenesis is happening. Spermiogenesis means what? There is specialized differentiation of these particular cells, and the sperms are coming out, leaving out the cell membrane and all the other residual bodies. That means not cell membrane, the cytoplasm and everything, extra cytoplasm is all removed off, because we need slim fellows who can go very fast. Okay, we don't want bulky fellows. Okay, we don't want extra weight. So all this extra weight is removed off and we get slim and trim sperms. Okay, so this process of conversion of cell body shape, this is called as spermiogenesis. Okay, the complete maturation of the sperm takes place in female genital tract, not in male genital tract. Okay, that's the beauty of it. The sperms are present in the male, but they are only capable to fertilize only when they enter into female genital tract. Okay, that is the complete maturation of the sperm. Now see the spermiogenesis. What happens in every cell? We have got, this is the till spermatid. Okay, what happens to spermatid? We have these particular structures inside cell. Inside cell we have cell body. Okay, this is cell nucleus. We have got acrosomal. What is this acrosome? This is nothing but the Golgi material, Golgi apparatus. You have studied cell, right? Golgi apparatus. Then we have mitochondria everywhere. See here, all mitochondria are there. And we have got axial filaments. Okay, so what they all do, they all accumulate together, they form a particular shape. What gives shape? If this is a viva question for you. We have a model here to show you a sperm, and we will ask you each structure is developed from where. Okay, nucleus gives the head of the sperm. Nucleus is giving head of the sperm. Golgi apparatus gives acrosomal cap. There is a cap. All the proteins accumulated here will accumulate inside this acrosomal cap, that will start sitting on the nucleus. So nice cap is sitting there. Okay, this is called as acrosomal cap. Then acrosomal cap is derived from what? Golgi bodies. Okay, Golgi apparatus. Then all the mitochondria, they will start accumulating around the neck region. Where is the neck region? How do I know where is the neck? Here what happens? We have studied that every cell contains centrioles, right? Centrioles are present. If you don't know, please go back and have a look at what are centrioles. So centrioles will come accumulate below the neck. Okay, below the head. This is the nucleus. Below the head there is one centriole, and the other centriole will be at the end. At the end, what it is doing, or what are these centrioles doing? They will start building up the axial filaments. That's what is giving cytoskeleton of the cell, right? It gives the skeleton of the cell, giving axial filaments. So all the filaments are developed, and all mitochondria, they will come and they will accumulate around this axial filaments, giving rise to a middle piece. Okay, so where we don't have a middle piece, the constricted area between the head and the middle piece, this is called as neck. What does neck contain? Neck contain only proximal centriole. What does middle piece contains? Middle piece contains mitochondrial sheath. Okay, and the rest of the axial filaments which are not covered by mitochondria, this is called as tail piece or a principal piece. What it is doing? Sperm is supposed to fertilize the egg, so it requires DNA, genetic material, which is inside the nucleus. It requires proteins which will break all the barriers that is present in the acrosomal cap. It requires energy to move, so mitochondria is here. It requires movement, that is coming from the tail. Okay, so this is a specific shape of the sperm. Okay, so now this is called as sperm or spermatozoa. Okay, so from spermatid to spermatozoa is called as spermiogenesis. Everyone clear with this?
Yes, ma'am.
Now see here, this is how the sperm is taken off, and you can see the residual body is left as excess cytoplasm is removed off, and this is the sperm. Now this is how we can have a look at sperm in a culture. Okay, if you just take out the semen and semen analysis, this is how the sperm we can see in a electronic microscopic view, the head and the tails. Now abnormal oocytes are there, abnormal spermatocytes are there. Sometimes what happens, we might see that primordial follicle with two oocytes. Okay, trinucleated oocytes. Usually we see dizygotic twins. Okay, when we see two follicles are with two follicle, single follicle with two oocytes are there. Okay, so we can see uh, twins, triplets. Okay, so usually what happens is abnormal oocytes and the sperms, they don't survive, they die after certain time. Okay, but if they are surviving, if they are fertilizing, it might be a problem. Oocytes, no problem, but sperms, if you see a sperm, it is these abnormal spermatocytes cannot fertilize the egg. Okay, only the best of the best will be chosen for fertilization. Okay, so see here we can see abnormal types of sperm. Okay, if there is abnormal oocytes like this, okay, this kind of problems can be seen. Okay, thoracophagous. So when both the fertilized fertilization is happening and the babies are born, but you can see that they are fused at some of the areas. See the abnormal sperms. This is in normal. These are electron microscopic views of semen analysis. We can see when the tail is completely curved. Okay, when there is no tail, when there is short tail. Okay, when double tail. Okay, double heads. You can see that there is no mitochondrial sheath. Okay, so these are the abnormal. So what happens is the semen analysis is done. For example, a patient comes to you, not to you, or for an infertility analysis, if they're going, they might have a good sperm count, but maybe abnormal spermatocytes are present, abnormalities are high. If the abnormalities are more than 10%, then the fertility becomes low. Okay, so that is also one of the reason why this is important to understand what is the normal and abnormality of sperm. So what are the questions? Oogenesis, spermatogenesis, and spermiogenesis. So that's all with this. Okay, are you understanding? Did you
Yes,
understand
ma'am.
this?
Yes, ma'am.
Yes, ma'am.
It will be bit, we are starting on with topics, will be going on in bit speed, high speed. Okay, so you will have